Hello, my name is Henry Gayhart, and in this video I will be discussing point mutations in DNA sequences. So for the sake of time, I will be under the assumption that if you're watching this video, you understand how we can get from a given DNA sequence to amino acids and codons. So in this, we are given an example template strand of DNA, which we are then going to go through the process of making RNA. And then after we have our RNA, we can divvy it up and find out which codons we have and which amino acids those codons actually code for. And after we have the codons, we can use this example to find out what a point mutation is, and we can find out if point mutations occurred in our example. So essentially, what a point mutation is a base substitution in the DNA. And this can occur through transition-based substitution, which is purine to purine, something like A to G or G to A, or pyrimidine to pyrimidine, like cytosine to thymine, or vice versa, or transversion-based substitution, which is purine to pyrimidine, or pyrimidine to purine. So the first point mutation is called a nonsense mutation. And essentially in a nonsense mutation, a base change causes a codon to change into a stop codon. As you can see in the example here, this base change from our original example DNA sequence from G to T, which is, by the way, a transversion base change, because it's G to T. Once we change that, this original codon now becomes UGA, which, if you look at your genetic code chart, is a stop codon. Next, we have missense mutations. Missense mutations is when a base change causes an amino acid to become a different amino acid. So in the example from ours, this G here goes through transversion missense mutation, because again, G to T, and this causes our mRNA to become AAA here, because TTT from our original DNA. So this TTG, which would be AAC, would code for lysine, but now TTT goes to AAA, which codes for ASN now. So a change in amino acids because of one single base. Next is silent mutations. This is essentially where a base change changes the codons, but both the original codon and the changed codon due to the mutation code for the same amino acid. So in our example here, a transition occurs this time because it's G to A, and this transition, as you can see, causes this codon here, CCG, change to CCA, which causes this to go from GGC to GGU, and both still code for GLY. So the same amino acid results from this mutation. And finally, we have read-through mutations. Read-through mutations are where a stop codon changes to a codon that codes for an amino acid. So in this example, we have the T, which changes to a C, which is a transition missense mutation, because we're going from a thymine to a cytosine. And this change right here in our mRNA causes UGG, which is, originally this would code for a stop codon. Now it codes for TRP. So essentially, when studying point mutations, it's important to understand that these mutations can cause serious changes to the DNA sequence, and thus the proteins that it codes for, but not always. In terms of silent mutations, the amino acid doesn't change even though mutation occurs. So not feasibly any changes to the protein occur, but in something like a read-through, a stop codon changes to a codon that codes for an amino acid. So massive elongation of a protein could occur, which could be very important and alter many things in the cell.